Hello friends, welcome back to another short video on the cancer biology or oncology. In this particular video lecture we shall be discussing about two major hallmark of cancer. So when I was studying these two effect of the cancer, those who are studying cancer, two effect you can see, one is the Warburg effect and second one is the Crabtree effect. So I was thinking about one question, if you take the cancer cell, if you take any cancer cell, so the cancer cell is acidic in nature. So when you're culturing the cancer cell, or if it is in the natural environment of its host, it shows acidity and the production of alcohol, ethanol, that is also very high. So the question is that what makes this cancer cell acidic in nature, and it makes difference response in corresponding to the adjoining cells, and it turns a little bit alcohol producing. So the first two aspects that we have to understand regarding the oncology, this particular cancer cell have a promotion and demotion of some biochemical pathway. Last decade, extensive and considerable research has been done on the cancer biology, and the scientists have found there is a promotion of some certain enzyme pathway and demotion of certain enzymes and pathway. So two effect has been studied by the Otto Warburg. He got Nobel Prize in physiology. Another is a Crabtree effect that was discovered by. I have written some statement regarding these two effect, and you will be answering me which statement is true or false. So Crabtree effect has been discovered by Herbert Grace Crabtree. Initially, I used to think like a Crabtree is like a crab is a cancer, zodiac sign, and tree is something to do with the proliferation. So it is somewhat similar that in cancer cells proliferation takes place, so that Crabtree effect that actually help in the proliferation, that is also true in that sense. But here the Crabtree means the name of the scientist. So let's come to the first point. So in both cases you can see there is a promotion of glycolysis, that is the basic fundamental concept, and demotion of the carboxylic acid cycle. Now the question is that why in both cases, or in the cancer cell or in the neoplastic cell, in the oncology, why the glycolysis is promoted or found in the higher rate, and the rate of OXPHOS pathway that occur in the mitochondria, so why that get reduced? One sentence answer is that there is a mitochondrial insult. That means when there is a cancer, other than the genetic mutation, the mitochondria of the cell first get insulted and damaged. So due to the partial damage of the mitochondria, there is a no chance that can lead OXPHOS pathway. So in the mitochondria, like if I take it as a mitochondria, so there is a damage in the mitochondria, so the cell definitely move towards the fermentation in the presence of oxygen. So oxygen is present, so they will move to the glycolysis pathway, and not that much oxygen is also required. You know, in glycolysis you don't need oxygen at all. So in the minimum amount of oxygen, so oxygen is present, and aerobic state they start fermentation. It is very, very interesting, and that is why cancer cell is told as a homologous to the yeast cell. So cancer cell is somewhat similar to the yeast cell. Like they have done research on the Crabtree also, Saccharomyces cerevisiae, Saccharomyces. So cancer cell and the yeast cell are very similar. Why it is very acidic in nature? In aerobic state means oxygen is present. They doesn't prefer TCA or carboxylic acid cycle. In case of TCA, they start fermentation. So due to the fermentation, they produce lactic acid. That is why the cancer cell turned acidic. Why this? This is because mitochondria is damaged. Another purpose of doing this one is very simple. They do not want to produce huge amount of ATP. So their purpose is to proliferate and cell divide and making the biomass. So what they should do? Suppose you have a carbon-carbon bonds and you have done oxidative phosphorylation. So what will happen? You have to break the bond and you have to go for a oxidative phosphorylation to produce the CO2. So now you can get excessive or sufficient amount of ATP. So in the meantime, what will happen? It is a catabolic pathway. So in the respiration, it is a catabolic pathway. But here what they are trying to do, they want anabolism. They do not want to destroy the material that they have consumed. So they will definitely prefer for the anabolism. So to promote anabolism, they will try to keep the carbon-carbon bond as it is. So that is why they will not tend to break it. So they will definitely go for a fermentation. So they do not want that much amount of ATP. So in the presence of oxygen, the lactic acid is produced along with our glucose. This phenomenon is known as the Warburg effect. Now what will happen? This lactic acid will go into the liver, and the liver will use ATP to get rid of lactate. So that is the entire pathway of the Warburg effect, where the fermentation takes place in the presence of oxygen. That is a simple definition. But in the Crabtree effect, what happens? You have an oxygen and you have a glucose. So in the presence of glucose and oxygen, the experiment was done in the yeast biochemist Saccharomyces cerevisiae. They found the Crabtree that presence of glucose and oxygen triggered the production of ethanol in the yeast cell. Similar effect has been observed in the cancer cell, that they can also produce ethanol in the presence of oxygen and glucose, because they are not interested to perform oxidative phosphorylation. So in this case, what is the main significance? Glycolysis is preferred over TCA cycle, OXPHOS pathway or TCA cycle. I have written some statement. So why acidity is seen in the cancer cell? This is due to the production of lactate or lactic acid. So another very interesting project you can run, those who are biotechnologists, you can use this cancer cell in a fermenter, and they could be a very good fermenting your lactic acid fermenter. So in place of a yeast that is used extensively in the biotech industry, you can also use the cancer cell as a acid producing, in the factory, in the vessel, you can use the cancer cell for the acid producer. Correct? Good fermenter. So this is the one of the extra use of the cancer cell. Correct? So cancer utilize energy from the cancer cell. Oxygen is not that much required, but if it is there, okay. Okay, now the statement is that cancer cell may delete some different enzyme or whole metabolic pathway. Is it true? Yes, it's actually the evident fact. In many cancer cells, they delete some pathway, and they sometimes delete entire enzyme. That is why in the TCA cycle, the enzyme that was required to run this cycle, all the enzymes, there is a disintegration or those are damaged. On the other hand, there is a promotion or the high fidelity, the production of the hexokinase enzyme. Hexokinase is very important for the glycolysis. So some set of enzyme in glycolysis goes up. So that is why one pathway has been selected during the course of evolution. The main purpose is that it has to proliferate, as you can see the cancer proliferation through the words. So their main function is to proliferate, not to produce energy for the body. Okay, so they thrive on the minimal enzyme, and they obliterate or they reduce the TCA cycle. That is true. And some tumor cells may show like a promotion of OXPHOS. That is little bit rare. Last decade, they have found some information regarding that one. Okay, the basis. What is the purpose of this Warburg effect? This effect can be used for the PET scanning, positron emitting tomography. So in this particular cancer, there is a biopsy through which you can test for the cancer. Positron emitting tomography scan is a method to detect which part of the body and those cells suffering from the cancer. So simply you use a radio level glucose. Like in the glucose you have oxygen, and you replace oxygen with the fluorine. We will discuss the different video. So simply you use the radioactive fluorine, and that will emit positron, that is the antimatter. So positron emitting tomography, so you replace the oxygen from the glucose, and in the cancer cell the glucose uptake goes very high. So that is why you can use this effect as a test. So these are hallmark of the cancer cell. These two effects are the hallmark of the cancer cell. Now they are saying this CC bond is preserved in the cancer. That is true. Okay. Now another last point they have given, the inverse Warburg effect. What is this? Same thing happens, but just to inverse it. Production of glucose from the consumption of the lactate. So you are consuming lactate and producing glucose. What is happening in the Warburg effect? You are producing lactate by the consumption of glucose. So inverse case, the reverse thing will be taking place. One very important value they have given. So rather than producing biomass via like a TCA cycle, in Crabtree effect, they produce like alcohols and aerobic, but the experimental research has been done and they have found the amount of glucose should be greater than 150 milligram per liter. So that is the important information I must share. So the amount of glucose should be more than 150 milligram per liter glucose and in the organism. In that case, you can see the favorable state in these two effects. So in that case, the Crabtree will be promoted. So this is the basic two hallmark of the cancer cell. You can see these two phenomenon, and whenever you are discovering the drugs, you can also apply this concept to create new drugs to apply the basic phenomenon, because you want to block some pathway. Suppose you are trying to block the pathway of a TCA cycle, and later you understood that this pathway is not at all present in the cancer. Suppose you want to create a blockage in a certain pathway in the cancer cell. First you have to identify what are the effects, what are the phenomenon, what are the pathway that is exactly happening in the cancer cell, what pathways are promoted, which pathways demoted or demotion takes place. According to that, after the complete knowledge, you can arrest cancer cells. Another thing that I have already discussed, you can use this cancer cell as a good fermenter. In the fermenter, like a biotech application, you can make a project out of it. Thanks for watching, and in the comment section you put some comments regarding what are the other effects you want to know. There are so many hallmark. This is the two most important thing I found when I was reading a question: why cancer cells show acidity? That's all. Thank you. Keep healthy, keep happy, and stay tuned.